Hello everyone and welcome to B3.2 in IB Biology, which is transport. Now let's get started. This topic is a bit confusing because there's a lot of different things, but we're mainly looking at the circulatory system in mammals and humans, and then also at plants and xylem and phloem. So let's get started. So first we're looking at capillaries. How are they adapted? So capillaries are the narrowest blood vessels, and they are in charge of exchanging materials between the blood and the internal or external environment. So they transport blood through almost all the tissues in the body. That's quite important. Therefore, they need a very high surface area for this exchange of materials. How is that achieved? Well, it's through their narrowness, right? So they're very, very thin, and they're also branched. So that provides a much higher surface area than if we just had few wide blood vessels. Then also the capillary wall is super thin. It's actually one layer of cells, so super, super thin, helping diffusion happen inside and outside. They also have a basement membrane. Here it's termed the basal lamina, and this just acts as a filter of sorts. And then thirdly, to increase exchange of materials, some capillaries have a lot of pores in their walls. So almost all capillaries have pores, but some of them have a lot of pores, and the ones that do are called fenestrated capillaries, so they have fenestrations, and this increases permeability when exchange needs to be particularly rapid in some organs of the body. Okay, amazing. So then we have arteries and veins. These are much larger, obviously. So arteries then become capillaries, which then become veins. So that's how the circuit works. And arteries carry blood at very high pressure because it comes from the heart, whereas veins carry a stream of low pressure blood because it comes from the capillaries and it goes back to the heart. Therefore, arteries have a thicker wall. As you can see here, it's really, really thick, but they have a narrower lumen, right? So the space is actually smaller, whereas veins have a less thick wall and the lumen is normally wider. This is not the best picture. You can't really appreciate the lumen difference, but that's normally the case. And then another good way to identify it, which you will be asked to do, is that arteries are almost always circular, whereas veins can be flattened, as you can see here. Amazing. So in terms of arteries, what's their wall made out of? What adaptations do they have? Well, they have several layers. They have the tunica externa, the tunica media, and the tunica intima. So the tunica externa is a tough outer layer, and it's mainly made of connective tissue with collagen fibers. Now, collagen fibers make arteries strong enough to withstand pressure changes. So that's quite important because the blood comes at very high pressure. Then the tunica media has smooth muscle and elastic fibers. So elastic fibers allow arteries to stretch and then recoil. And what that does is it squeezes the blood in the lumen, helping it move forwards. And then the smooth muscle helps change the diameter of the lumen, allowing to adjust blood flow to different tissues. So if you want more blood flow to an organ, then you can make the lumen larger through changing the smooth muscle contraction to that artery. And then the tunica intima is a thinner layer. It's the lining of the artery, right? And it's made of smooth endothelium. Great. So then veins. So arteries, as we said, they have blood at very, very high pressure because it comes directly from the heart. Then that's distributed to the organs through the capillaries, but then that has to go back to the heart via veins. So again, veins collect blood from all organs and bring it to the heart, but there's no pulse, right? Because this is happening continuously. The capillaries are feeding to the veins continuously. So the wall contains far fewer elastic fibers and smooth muscle cells than artery walls. Therefore, they're less thick. The problem is because the blood is at a low pressure, there's the chance of backflow, meaning it could go back to the capillaries. And that's why veins have pocket valves. So if blood starts to flow backwards, pocket valves fill with blood. You can see that here, right? It's meant to go up, but if it goes down, then this will start to fill up and these flaps will close the lumen, meaning it will not be able to go back. However, if the blood flows forwards, as it should be, it pushes the flaps to the sides of the vein. That's how you can see it in the image here, allowing the blood to flow through. So that's quite a clever system that the veins use. In this case also, what helps blood flow through veins? Muscle contraction. So muscles that are close by contract, helping squeeze the blood to move through the veins. Great. So coronary artery occlusion. This is the last thing for SL on the circulatory system. So what are coronary arteries? Okay, so the aorta, okay, carries blood to all organs apart from the lungs. And the right and left coronary arteries branch off from the aorta and basically supply blood to the heart itself. The heart is a tissue which needs blood, so it's the coronary arteries that supply blood to the heart. So the coronary arteries can often become narrowed or totally blocked, and that's called occlusion, and by fatty deposits, right? And that's called an atheroma or a plaque. I know there's a lot of words here to remember, which is annoying. But these deposits, as you can see here, build up in the wall of the artery, and cholesterol plays a part in this. And if these deposits become impregnated, so covered by calcium, as you can see here in a slightly darker color, this can harden the artery. And that triggers the formation of a blood clot. That's called thrombosis, okay? When that happens, a clot can actually block the flow of blood, preventing the heart from contracting because the heart is not receiving blood to oxygenate its tissue, and that can lead to a heart attack. Okay, so just to make it clear, it's actually very rare for the plaque itself or the fatty deposits to cover the entire artery, but a lot of times, again, when calcium covers it, that hardens the artery, triggering a blood clot, thrombosis, which then actually blocks blood from going through. Okay, so what are some risk factors? Well, quite obvious, right? Hypertension, smoking, obesity, age, sedentary lifestyles, alcohol overconsumption, etc., etc. And all of the conditions that are associated with this blockage are named coronary heart disease. So hopefully that sheds some light. Maybe go back and review all the terms because there's a lot. I understand that. Okay, so moving on to plants. So how does water flow from roots to leaves? Through xylem. So xylem is the tissue used by plants to transport water. And how does that work? Well, because it's going against gravity, right? Well, transpiration, if you remember from the previous topic, causes water evaporation from the cell walls of spongy mesophyll cells and diffusion out through the stomata. So hopefully you remember that. And that's because of humidity differences in the air and the leaf, right? So stomata have to open for air to pass through, but that also makes water leave. So then what happens is that water from the xylem is drawn to cell walls in the leaf through adhesion. Remember, water adheres to polar substances. And what that does is that creates a tension, right? Because water is being drawn out of the xylem through adhesion. That creates capillary action, it's called. So it's a pulling force. As long as there's a continuous column of water in the xylem vessel. So that's important. There needs to be a continuous column of water in the xylem. So if a little bit is being pulled to the leaf at the top, that creates a tension which is carried downwards, and then that's drawn to the root actually, which is strong enough to move water upwards. And the continuous column of water is ensured by cohesion. Remember the property of water, which means that water it sticks to each other, right? Water molecules stick to each other. Okay, hopefully that makes some sense. Again, any questions, make sure to leave them in the comments and I'll answer them as best as I can. So what are some adaptations of xylem vessels? Okay, so xylem vessels are formed by columns of cells, although it doesn't really seem like this is a cell, but well, these were cells that then have been emptied out. So basically the cell wall between the cells, as you can see here, is empty. There's no cell wall here. And the material, the contents of the cell are also incomplete or completely absent. And this creates these continuous tubes, meaning water can flow through very, very well. Walls are normally also thickened and impregnated with lignin. This is this thing you see here, this ribbon of sorts. And that keeps walls from collapsing under tension and under low pressure, which are circumstances that the xylem finds itself in constantly. So lignin is actually permeable to water, it's hydrophobic, but there are always gaps. They're called pits, as you can see here, right? These holes, which allow water to enter and exit the xylem. And this is a micrograph of the xylem, so you can see how it looks like in real life, and you might be asked to identify this. And it's very characteristic because of these rings, right? This ribbon, and this is lignin that you can see here in the tube. Okay, so now you have to be able to identify this in a micrograph, so in an actual image. All right, so first you'll see that you have these groups, these bundles. They're actually called vascular bundles because all of the vasculature is found here. The easiest way to remember is that the xylem is the largest circles, right? So remember, xylem is the transport of water from roots to leaves. Then you have the phloem, which are further away from the center normally, and they're much smaller. And that is for the transport of sugars from leaves to roots, mostly. There's also other pathways. And then the cambium is what's in between the xylem and phloem, and it's actually tissue that produces more xylem and phloem. Then we have the pith. So the pith is almost everything, right? The whole center, the really, 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 really big cells. And that's just for the stem to be bulked out, for it to be bigger and be strong, right? For it to be able to stand upright. Then we have the epidermis. That's the most outer layer. It's for waterproofing and protection. And then the cortex, slightly more inside than the epidermis. The cortex is, again, for support and also for photosynthesis. So the stem can actually do some photosynthesis. But most importantly, I would remember that the xylem is here and the phloem is here. That's what will be asked most of the time. And then we have the roots in a similar manner. You have to remember, well, in this case, the phloem and the xylem is found in the center. And again, the xylem is the bigger circles. And a lot of times they'll be stained red, and that's because of lignin. Lignin can stain it in a reddish color. But don't trust that, because in IB in the papers, it's black and white. Then the phloem is what's outside of the xylem. So it forms kind of like a star or an X. So the X is the xylem, and then what's around the X is the phloem. Again, then everything else is the cortex. And then you have the epidermis and endodermis. So the endodermis is the circle that surrounds the xylem and phloem. So again, just look at this for a while, memorize it. Should be quite simple. Now, for higher level. So, tissue fluid. What is this? Well, plasma, okay, is the fluid in which blood cells are suspended. So it's kind of the liquid part of blood, okay? This red thing that you can see here, or pinkish thing that I'm highlighting. It's mainly made out of water, but it also has glucose, amino acids, minerals, etc. Now, the capillary wall, as we talked about before, has pores in it, right? It's easy for things to go in and out, meaning that actually the capillary wall lets plasma leak out into the spaces between cells in the tissue. This then gets called tissue fluid, okay? It's basically the same thing as blood plasma, except it doesn't have the really, really big proteins, because that cannot leave through the wall. They're too big. So this is continuously released and reuptaken by the capillaries. So capillaries close to the arteriole release it because the blood is at a high pressure, and then capillaries closer to a venule reuptake it since blood pressure is much lower inside the vein. So again, tissue fluid is going to contain all of these nutrients except for large proteins. And then cells can actually absorb these substances as the tissue fluid is surrounding them, right? So they will uptake oxygen and glucose, for example, and they will excrete carbon dioxide and other waste products. Then these re-enter the blood plasma, and that's then taken away by the veins and purified. So the CO2 is purified by the lungs, and then the waste products by the liver or the kidneys. So this actually is very, very important. Okay, think about it. We always talk about how ventilation happens to get oxygen, but how does that actually have an effect on cells? Well, it's through this, right? It's through blood plasma leaking out, which then allows exchange to happen between cells. Now, a small part of the tissue fluid actually, instead of being taken back up by the capillaries, it's drained into the lymphatic system. So lymphatic vessels also have permeable thin walls, and once inside the lymph, it'll be called lymph instead of tissue fluid. So remember, blood plasma, tissue fluid, and then lymph, basically the same thing. Then these lymph vessels, again, join to form wider lymphatic vessels. And at the end of the system, there's the left and right lymphatic ducts, which then merge with the subclavian veins. So actually, it is returned to the blood system eventually, right? It just takes another path there. We've looked at the different types of veins, capillaries, arteries, but now let's look at the actual circulatory system. How does it work as a whole? Well, in mammals, here on the left, first, mammals pump blood to the lungs over here to be oxygenated, but it does so at a low pressure. Why? Because otherwise capillaries would burst, and the alveoli as well. But then after this, it's at a very low pressure, meaning it can't go to all of the other organs, right? For example, to go to the brain, it would need to go against gravity, so that would require a lot of pressure. So instead, it returns to the heart, as you can see here, to be repumped. And then it's repumped to the systemic circuit, right? And this takes it to all the organs through capillaries and then returns it to the blood. So basically, The left side of the heart receives oxygenated blood and pumps it to all the organs, apart from the lungs, at very high pressure. This blood then returns to the right side of the heart, this time deoxygenated, which then pumps it to the lungs to be oxygenated. This is not the case for all animals, so this is double circulation, right? Since the blood passes twice through the heart to make a full circuit. They're called pulmonary and systemic circuits. But for example, for fish, here on the right, fish actually pump blood to the gills over here for oxygenation, and then that continues to flow to other organs, after which it comes back to the heart. So it's a single circulatory system, and it's because it doesn't need to be at a low pressure to go to the gills, unlike for humans where it does need to be at a low pressure to go to the lungs. Okay, so now for the actual tissues of the heart, it's important to understand the adaptations. So we have the ventricles, right, which are found at the bottom. They're quite large, thin chambers. So they have very strong muscular walls, which you can see here. Why? Because they need to generate very high blood pressure when it contracts. Why? Because this is being pumped out to the arteries. So ventricles pump out to arteries. And remember, arteries have blood at very high pressure. Then we have the atria, right? So the atria are over here, and atria are actually chambers with thinner muscular walls. Why? Because they're collecting blood from the veins. So they actually only need to pump the blood to the ventricle, which is right there. So it doesn't need a very strong force. Then we have atrioventricular valves. So these are found here and here. So between the atria and the ventricles, as the name indicates, and these close once the blood is in the ventricles to prevent backflow to the atria when the ventricles contract. Similarly, the semilunar valves, which you can see here, between the ventricle and the artery, close to prevent backflow of blood to ventricles when they're relaxing and the blood is in the artery. Okay, so then these walls that we're talking about between ventricles and atria are formed by cardiac muscle. So cardiac muscle has branched cells, and there's connections between the plasma membranes of adjacent cells. That's very important because it allows electrical signals to be propagated throughout the wall of the heart, and that allows coordinated contractions. So it allows a contraction to stimulate other contractions very, very quickly. That's again through these connections between plasma membranes. That means that the heart can contract without stimulation from motor neurons, and that's unique. It's called myogenic contraction. And also, the heart has a pacemaker, so that's called the sinoatrial node. It's found in the wall of the right atrium, so somewhere over here, and it sends an electrical signal initiating each heartbeat. So the membranes of their cells of the sinoatrial node are the first to depolarize in every cardiac cycle, and that activates adjacent cells, again, through this connection between different cells of cardiac muscle. And then finally, the septum is the wall of, it's here, right? It's the wall of the heart between ventricles and atria on the right and the left side, and prevents blood from mixing between the two sides, which is very important. And then finally, coronary vessels. We talked about them before in standard level coronary arteries and veins. They carry oxygenated blood from the aorta to the heart wall and collect deoxygenated blood from heart walls. And again, it's important because coronary heart disease is a very prevalent disease which affects a lot of people. Great. So then in terms of the cycle, how does this work, right? So this is a very common diagram. You can see here atrial pressure and ventricular pressure. So this is when the atrium contracts, right? It increases pressure by a little bit, but remember, it has a thinner wall. It doesn't contract as strongly. After which the AV valves close so that it doesn't flow backwards. Then the ventricle contracts, leading to a super high increase in pressure because, again, it has those really thick walls and it needs to pump it at a really high pressure. And then that causes the ventricle to go up like crazy in terms of pressure, which then makes it flow to the aorta, right? Meaning the aortic pressure also becomes really, really high. Then the semilunar valves close, remember, so that it doesn't backflow to the ventricle. The ventricle decreases in pressure. And then the atrioventricular valves open so that, again, afterwards, the atrium can contract and pump it to the ventricle. So, important. Obviously, the sinoatrial node initiates every turn of the cycle. And then when a chamber contracts, the pressure of blood increases, obviously, and that's called a systole. Then, when pressure drops after contraction has stopped, that's called a diastole. So those are two important terms to remember. Okay, and then the last two things for higher level, which are about plants. it's about transpiration again. So we talked about how there's these tensions, this capillary action, but sometimes transpiration is not occurring. Why? For example, at nighttime, because photosynthesis is not happening, or there might be very high atmospheric humidity, so water is not transpiration is not happening. That means that water would not be drawn from the root automatically because there's no tension. So what happens? Well, what happens in this case is called root pressure, meaning root cells actually pump minerals into xylem vessels, so from yellow to blue, by active transport. And what that causes water to move to the xylem via osmosis because of the gradients of minerals, right? And that pushes the sap upwards. Quite simple, but that's what happens, right? Minerals are pumped to the xylem so that water will flow consequently. And then finally, the phloem. Okay, so we haven't talked about this. So the phloem serves to transport sugars in a plant, right? So sugars are transported from sources to sinks. So for example, a common source is leaves, where photosynthesis happens, and a sink is roots, because roots need glucose, but they can't produce it themselves. So sieve tubes provide the channels in the phloem, and they develop from columns of cells, which become connected by large perforations in their cell walls. As you can see here, these are called sieve plates. So in the xylem, they're completely lost. In the phloem, they're perforated, and this allows contents to flow through the phloem. Then to actually load and unload sugars at sources and sinks, active transport is required. But these cells, right, the sieve elements, can't do it in enough quantity. Why? Because they have very limited cellular components. So they have these companion cells, which are adjacent, and they have a lot of mitochondria. So companion cells also have cytoplasmic connections with the sieve elements, which are called plasmatosmata. You can't really see it in the image, but they would be over here, right? These connections. And then sugar unloading and loading through active transport also makes water be drawn in. So for example, during loading, you're putting sugars into the phloem, meaning water will go in through osmosis. And then when you unload at a sink, water will leave via osmosis, right? And this creates pressure differences, which drives the flow of sap from sources to sinks. So that's how we make sure that phloem flows in the correct direction, through the osmosis of water and the differences in pressure that creates. Hopefully that's clear. I know this topic is quite complex. Okay, let's do a couple questions. So first, which of the following is not a contributing factor of atherosclerosis formation? I'll let you think for a sec. Pause now if you want to think, but in 3, 2, and 1. The circulation of unsaturated fats in the bloodstream. So if that is happening correctly as it should, there should be no problem, right? It's when they deposit to the actual artery. So high blood pressure does not help. The hypertension, right? The buildup of atheromas, that's what I said. So when they actually build up in the artery, that's really bad. And then also when the artery is damaged, that can also create blood clots and be very detrimental. But the actual circulation of fats under normal circumstances should be fine. Great. Okay, which letter identifies the phloem in the following micrograph of a root? So hopefully this is straightforward. Again, 3, 2, and 1. It's B, right? Remember, the bigger ones are the xylem, and again, they're tinted reddish. And then what's around the star is the phloem. So B is the phloem, and then C would be the xylem. Okay. And finally, which is not a feature of the sinoatrial node? This might be a bit more tricky. Again, pause now. And in 3, 2, and 1, it's D. It doesn't occupy the area between the right atrium and the right ventricle. It's in the wall of the right atrium, right? It does act as a natural pacemaker. It is formed from a group of highly specialized muscle cells, remember, it allows them to depolarize very, very quickly. And it sends out a wave of electrical activity, which causes contraction. And that propagates to the whole heart, so to both atria and ventricle. So that's a common misconception. It's to both. So it's D. Okay, so that's a wrap. I know this topic is quite complex and there's a lot of concepts that are mixed together, but hopefully that was clear. If you liked this, please do like this video, and I'll see you next week for the next topic. Again, any questions in the comments, and hope you have a good week. Bye-bye.